Hello, good morning everyone. I'm Nurul. Today, I'm very delightful to present our research by the title of Distribution of Black Shank Disease on Temanggung Tobacco and Environmental Factors Affecting Disease Development. I would like to divide our presentation into 5 parts. The first is introduction, the second is objective of the research, the third is methodology, the fourth is results and discussion, and the last part is conclusions. Here is the background of this research. Temanggung tobacco is type of dried slices tobacco. It is a main component of clove cigarette. Temanggung tobacco also has specific properties. They are aromatic and have high nicotine content about 3 to 8%. Although Temanggung tobacco rolls as a major component of clove cigarette, the production of Temanggung tobacco only meets 30% of cigarette factories' requirements due to low productivity. There are at least 2 factors affecting low productivity of Temanggung tobacco. The first is invasion of soil-borne pathogens such as Phytophthora nicotianae, the causal agent of black shank disease, and the second is degradation of land fertility. However, there is no information about the distribution of black shank disease, which could cause difficulties in managing the disease within the field. Therefore, our research aimed to determine distribution of black shank disease within tobacco plantation in Temanggung and identify factors affecting disease development in the field. Let's move on to the methodology of this research. Our research was conducted by a survey on 53 sampling points determined by land system method. It spread on 13 sub-districts and 43 villages, with various elevation ranged from 500 to 1400 meters above sea level. On 53 locations, we did observe incidents of black shank disease by recording number of infected plants for each point. Then it was classified into 5 classes. Very low is less than 10%, low is ranging from 11 to 30%, moderate is 21 to 30%, high is 31 to 40%, and very high is more than 40%. In addition to observation of black shank, we did also take soil samples to determine soil nutrient contents. While taking the soil samples, we also interviewed the farmers to achieve information on the history of the land and cultivation methods that they are practicing. This is a map of Temanggung district. The black dots indicated locations of 53 sampling points. Before going to the distribution of black shank disease, I would like to show the symptom of black shank in the field. Tobacco plants infected by Phytophthora nicotianae would perform suddenly wilting and yellowing leaves like the picture on the right. In addition, the lower stem is also rot and necrotic. When the stem is cut, it would appear blackened pith with distinctive discs along necrotic stem, like the picture on the left. Then, how is the distribution of black shank disease in Temanggung? Our survey revealed that black shank disease was found mostly in the lower elevation areas in paddyland, and we found that the highest incidence could reach up to 44%, which is in sampling point of 39. We didn't find black shank on elevated location. It might be related to the environmental condition when we did survey. At the time, the condition was very dry as there was no rain for 3 months. That's why none of black shank found. On the other hand, in paddyland, they have a good irrigation system, causing soil becomes more saturated. In that condition, wet, saturated soil and warm soil temperature is favorable for black-shank development. Our survey also revealed that organic matter content within soil of tobacco land also ranged from very low to moderate. As tobacco land in Temanggung experienced severe land degradation, most of topsoil was degraded. Consequently, they lost their organic matter. This condition influences soil microorganisms population in habits within soil. When these organisms decreased, the pathogen might be able to grow extensively and cause disease on the host plants. Our results also revealed that organic matter has negative correlation with black-shank incidence, which means if the organic matter is low, the incidence of black-shank is high. According to the results, we could conclude that black-shank disease was found on paddyland characterized with wet soil. It also relates to low organic matter. Some areas with very low organic matter was found to have high incidence of black-shank disease. In addition to low organic matter, soil temperature, moisture, and pH also affect development of black-shank disease. Finally, we would acknowledge Bapeda Temanggung for providing funding for this research and also tobacco farmers for their cooperation during conducting this research. Thank you for your attention.